Thank you everyone for being here. I'm really excited to share some of the research I've done for my master's here at SIU, looking specifically at the occupancy and ecological influence of semi-aquatic mammals here in Southern Illinois. All right, so as most of you are probably already aware of, aquatic systems are important for a lot of different reasons. Wetlands specifically are really good at nutrient trapping as well as mitigating the impacts of flooding and erosion across the landscape. They also provide habitat for a lot of different wildlife and plant species and therefore support biodiversity. You also have these riparian areas, which provide similar benefits in terms of those nutrients and biodiversity, but these can also specifically act as movement corridors for certain wildlife species that may use them. Within both of these systems, semi-aquatic mammals play a couple of different key roles. So the American beaver there on the left and the muskrat on the right are often described as ecosystem engineers. And that's because of the individual impacts that their behaviors have, not only on the landscape, but to the resources available to other species. So beavers, for example, create dams that has cascading effects, and then muskrats can increase habitat heterogeneity through their foraging. You also have the American mink, they're on the bottom left, and the river otter on the right. And these are the top predators in these systems. And as such, they're usually sensitive to water quality and require specific habitat requirements. Therefore, their presence is often associated with some indicator of the overall health of that habitat. So these species are important, and they can be somewhat difficult to find. They're elusive and spend much of their life in the water. So traditional methods, besides actually handling these animals, rely on looking for their tracks in scat. This requires not only good observer skill and being able to identify the actual track or scat to the species, but it requires a bit of luck as well, just because these tracks and scat are often subject to, environmental conditions, and they can just get washed away in rain. But thankfully, technology is awesome, and we do have other methods. So camera traps are used for a lot of different wildlife species and can be used for semi-aquatic mammals as well. And then excitingly, you do have environmental DNA being used for semi-aquatic mammals now. This is traditionally a fully aquatic approach, but again, given that semi-aquatic mammals are semi-aquatic, it's being branched out to use for these species as well. So semi-aquatic mammals are well researched, but there may still be some general gaps in our understanding of their distributions. If you look at a lot of the previous research done, it usually focuses on their distributions in either lentic or lotic environments. So if you're not familiar, those lentic water bodies are those that are non-moving, so such as your ponds and lakes, and then lotic are your moving water bodies, such as streams and rivers. And again, most studies that address semi-aquatic mammal occupancy rarely do so in looking at both of these types of water bodies. And then as far as I'm aware of, there's been no independent consideration of how terrestrial versus aquatic factors influence where you might find these species. This is simply important because these areas are managed for different goals and purposes. So for example, for aquatic systems, they're often managed for water quality and or the vegetation that you might find there. And for terrestrial systems are often managed for land use. Between terrestrial and aquatic systems, beaver dams tend to blend the two together. So as I mentioned earlier, beavers create dams, which then slows that flow of water, creating more of this lentic environment. You can see in the photo here, it kind of balloons out, and that helps promote organic soil. That soil then promotes the growth of different aquatic vegetation, then in turn creating new habitat. This creation of habitat from beaver dams is well studied and has actually been shown to increase the species richness, which is simply the number of species at a given site in a variety of communities. So for example, it's been shown to increase species richness in water birds, in different vegetation communities, amphibians, and even scaling all the way up to increase the species richness of terrestrial mammals. So building these beaver dams are perhaps the most notable behavior of beavers, but they do have other behaviors that can be considered engineering and impact the environment. So of course, to actually build these dams, they do chop down trees, not only to build dams, but build their lodges and to forage as they eat the cambium. They also build these beaver runs in which they dig. Just allows more basically transportation and water. That way they have to cover less ground to access those resources. And both of these behaviors do have effects on the environment. Cutting down trees not only adds more woody debris to the landscape, but it also changes the physical structure and composition of that area. And these beaver runs specifically have been documented to retain water in periods of drought, whereas other water bodies that don't have these tend to dry up in periods of drought. So they do have, these effects on the environment, but it's unclear if they scale up to influence species diversity in the same way that beaver dams do. As I mentioned, muskrats are also an ecosystem engineer, and there is a pretty wide body of research showing these different effects that they have in increasing habitat heterogeneity, mostly through their foraging. But again, similar to those other beaver behaviors, it's less clear if and how those effects scale up to influence species diversity. So far, all of the effects of semi-aquatic mammals that I've talked about have been fairly positive. That's not necessarily the case. If you're not familiar with this animal, this is the nutria, which is an invasive semi-aquatic mammal here in North America. They are larger than a muskrat, but smaller than a beaver. They have these distinct white whiskers, and I think if you kind of catch them at the right angle, they look like a small capybara. So as I mentioned, they are non-native here, they're invasive, so they're native to South America. They were introduced to North America for both the fur trade and biological control of vegetation, which always turns out great. Wild populations were established either through escapee individuals or when the fur market declined, a lot of people just let these animals out in the wild and they established populations. Nutria are invasive specifically because of their voracious appetite. They can consume up to around 25% of their body weight daily, which can quickly lead to something that looks like this photo here, where you see this normal marsh in the background, and then all this brown is where nutria have completely denuded, or in other words, eaten the entire vegetation layer through the roots. So that soil will just be subject to erosion, and then that area will become less productive. Like many other invasive species, nutria are very hard to get rid of once they're established. There's this case study in the Blackwater National Wildlife Refuge in Maryland, in which nutria were introduced there in the 1940s. This area has a lot of this freshwater coastal marsh habitat that nutria love. So sure enough, the population boomed, and by the 1990s, you had this massive wetland loss in these areas. So the federal and state agencies got together, and they formed this Chesapeake Bay Nutria Eradication Project in 2002. Given that their goal is eradication, they did try a lot of different methods to get rid of nutria, including doing these boat surveys. They built these floating raft platforms that mimic the natural feeding platform of nutria. So nutria would jump up there, and they needed to have a hair snare or even a trap to remove that individual. They also formed this Maryland Detection Canine Program, in which they trained these shelter dogs to detect nutria scat. And sure enough, after 20 long years of completely internal work within that government agency, they were able to confirm the eradication of nutria from that Delmarva Peninsula. When putting this presentation together, actually, I saw that there was a sighting of nutria on that peninsula in 2024 last year. So all that to say, it's very hard to get rid of nutria once they're established. While all that work was completely internal, other agencies tend to take a different approach and encourage the public to get involved. So the U.S. Fish and Wildlife Service put out this article earlier this year, 5 invasive species you can hunt, catch, and cook. And if you go to nutria.com, which is run by the Louisiana Department of Wildlife and Fisheries, you can even find a nutria gumbo recipe, all to encourage the public to kind of get involved and kind of help get rid of some of these individuals. So looking back, taking a step back, and looking at all states of where nutria have been or currently are, you can see it's just over 22 states where nutria have been or are. And I would like to point your attention to a small little blob close to where we are here in Southern Illinois. And sure enough, there was a 2013 sighting of a nutria up in Rend Lake by the Army Corps of Engineers. Sure enough, that's not the only sighting of nutria in Southern Illinois. The Illinois Department of Natural Resources has actually received numerous anecdotal reports of nutria in the area since the early 2000s. But looking at these photos, you can see it's pretty easy to confuse a nutria with a beaver or a muskrat, especially if it's off swimming in the distance, or in this case, on your trail camera, in which you might think you have a nutria. These are trail camera photos that I've received, in which instead it's not a nutria, it's a beaver and possibly a backlit possum even. But sure enough, in 2022, two nutria were actually trapped in a beaver trap in Pope County. So that really prompted the need for the IDNR to kind of investigate and see what the real status of these species are. Taking a step back and looking at Southern Illinois as a whole, there's numerous lentic and lotic water bodies. We have a lot of large lakes and small ponds and big rivers and small streams. And then additionally, if you look at some of the beaver research that's been done in the area, most researchers note that they've found very few beaver dams in this area. It's not necessarily known exactly why that is, but altogether, beavers are not necessarily synonymous with beaver dams in Southern Illinois. So taking that as a whole, Southern Illinois makes a really great area to address some of the gaps in our understanding that I've talked about, specifically this difference between aquatic and terrestrial factors, lentic and lotic water body types, and this possible influence of beavers with and without a dam. So that's exactly what I did. I identified if aquatic or terrestrial factors are more important to semi-aquatic mammal occupancy, and then determined which of those factors are going to have the greatest influence. I also assessed the possible impacts of beaver and muskrat presence on terrestrial mammal diversity, specifically, again, looking at how that may change with and without a beaver dam. And then finally, I determined the status of nutria in Southern Illinois. And I did that by comparing some different survey methods to try and see which may be the best to actually finding these animals. To address all of these questions, I randomly selected 67 different sites across the lower 4 counties of Southern Illinois, proportionally stratified by both water body type and size, according to what was available in the landscape. So you can see I have both lentic and lotic water bodies, and then 5 different size classes within each of those types. For lentic water bodies, again, those that are non-moving, I stratified by the area of that water body. And then for lotic water bodies, those that are moving, I stratified by the stream order. At each of these sites, I conducted a couple of different survey methods. So for all semi-aquatic mammal species, I went in with a technician, and we went and conducted 2 independent sign surveys at each of those sites. So we flipped a coin, determined the direction that we would walk, and then walked 200 meters along the water body's edge, recording all tracks and scat. And then right at the vertex of those sign surveys, we placed a single camera trap within 5 meters of that water's edge, and then let it do its thing. We checked these sites bi-weekly for 3 times, which resulted in 6 independent sign surveys and about 4 weeks of camera trap data for each site. Then specifically just for nutria, we actually brought in some of those detection canines from that Maryland detection canine program, in which we had 2 canines and their handlers visit each site once. They did their own thing, kind of looking around, sometimes even within that water body, looking only for nutria scat. And then Madison Sikorsky and Adrian Stanley braved the summer heat and went out and collected some eDNA samples. So at each of those sites, they went in and collected 3 different samples in which they filtered 5 liters of water, and then they were able to take that filter that might have any genetic material and dry it back here on campus. And then they shipped those off to an external laboratory for qPCR and metabarcoding analysis. So moving on to my first research objective here, identifying this difference between aquatic and terrestrial factors. To do this, I used occupancy modeling, which is just a form of modeling of species distribution and where you'll find them. And this model focuses on having repeated visits to a site to not only allow a researcher to estimate where the probability that you might find a species there, but also the probability of the species being there, and then the probability of actually detecting that species. So of course, if you go to a site and you see the species, it's there. But there's also the possibility that if you go to a site and you don't see the species, it could be there that it's just not there in reality, or that you just missed it. So occupancy modeling accounts for that imperfect detection. And given that it requires these repeated visits to a site, I'm only using sign surveys and camera traps because those had repeated visits. There are a lot of different flavors of occupancy modeling, if you will. I'm using the multi-method occupancy. This model kind of has its root in this multi-scale occupancy model, in which the researchers were interested in using multiple survey methods to estimate 2 different scales of occupancy probability. I instead used an approach more similar to Berry et al. 2021, in which they still used multiple survey methods, but instead had those contribute to just one estimate of occupancy. And you can see that in the equations to the left. I'm still able to retain a method-specific detection probability, but both of those contribute to the one estimate of occupancy. To actually look at any habitat influences on occupancy, I use the standard logit link function to attach my covariates. And then I built these models in Nimble. And then there's some of the model specifications. I started with uninformative priors, used a burn-in of 5,000 iterations, 3 chains of 20,000 iterations, and thinned by 1. So to actually address this possible difference between aquatic and terrestrial factors, I performed a model selection. So first, I started off by determining my top detection model for each species. And then I considered just my aquatic covariates, which were the aquatic type and size of each of those water bodies. I considered different additive effects and interactive effects to then determine my top aquatic model. I then did the same thing with my terrestrial covariates, which were the proportion of land cover type, distance nearest road, and elevation. And for that land cover, I did consider different buffer sizes for each type to determine the best buffer size, and then use that in each model. Similar to aquatic, I considered different additive, interactive effects to then determine the best terrestrial model. I then just wanted to go one step further and combine those two top models to be able to then compare what that would look like and how those perform to the top individual. And to actually perform this model selection, I use the R package LU, which uses a leave one out cross-validation. So quick overview of the data I collected. In total, I had 402 sign surveys, over 1,800 trap nights with cameras. That yielded over 103,000 photographs, 75,650 of which were empty. And that's somewhat reflected in this bar chart to the right, which shows the number of sites with detections from each survey method for each species. It's pretty clear looking at that you can see that assigned surveys detected these species far greater than camera traps did. Moving on to the results from that model selection, I have a table of the top aquatic and terrestrial model for each species, along with their associated LU-IC score. LU-IC is interpreted the same as AIC, in which a lower value indicates a more supportant model. But unlike AIC, it doesn't have that delta 2 value that signifies a difference. Therefore, I'm just using the relative differences in these values. All right, so we can see that the top aquatic model was more supported for beaver, otter, and mink, whereas the top terrestrial model was more supported for muskrat. Bringing in that combined model, sure enough, the combined model is more supported for all species across the board. But again, looking at those relative differences kind of provides a bit more information. Specifically, beaver and otter, you can see that the aquatic model was more supported than the terrestrial, and there was a fairly large difference between those two. But then when you look at the top aquatic to the top combined model, it's a pretty minimal difference. And overall, what that suggests is that this terrestrial model isn't adding much predictive power in predicting the occupancy of beaver and otter. Looking at muskrat and mink, it's a bit of a different story in which the top aquatic and terrestrial model is different, but the actual value differences between those two is pretty small. And then when you look at both of those to the combined model, it's a much bigger difference. So to me, that suggests that both of these top models are likely contributing equally to that combined model. Moving on to the specific model coefficients of those combined top models for each species, I consider both the 85 and 95% credible interval. Anything significant or non-overlapping 0 at the 85% credible interval was interpreted as somewhat likely to have an influence, and anything significant at the 95 was considered strongly likely to have an influence. So for beaver here, we have distance to road, as well as water body type and size in the top model. And here we can see that both the largest and second to largest size classes were strongly likely to have a positive influence. And the intercept term here, which simply represents small lentic water bodies, was somewhat likely to have a negative influence on their occupancy. Moving on to muskrat, here we have distance to road, as well as the proportion of crop within a 500-meter buffer, and the interaction term of those two, and then water body type. And we can see that both distance to road and loadic water bodies were strongly likely to have a positive influence. River otter actually had the same top model as beaver, so distance to road as well as water body type and size. And here we can see that just the largest size class was strongly likely to have a positive influence, whereas the second to largest was somewhat likely to have a positive influence. Last but not least, the American mink. Here we have the proportion of wetland within a 250-meter buffer, as well as distance to road, and then an interaction term of water body type and size, in which size was simply condensed down into 3 different categories. And sure enough, nothing was strongly likely to have an influence, but instead distance to road, as well as this interaction term of water body type to the largest size classes, was somewhat likely to have a positive influence. So what does all that really mean? Sure enough, I did find that the combined model provides the best predictions for all semi-aquatic mammal species. That's fairly intuitive, given that these are semi-aquatic mammals and they use both. But again, those relative differences provided a bit more information. Specifically, that it seems that these aquatic features are going to be more important for beaver and otter, whereas both seem to be equally contributing to predicting the occupancy for muskrat and mink. I also found some interesting differences between water body type. Specifically, that type wasn't influential for otter and mink occupancy. While that may not seem interesting, it's really suggesting that if you're not more likely to find these species in one over the other, they're likely using both. And I think that does have some implications for previous research as well as future research and looking at these species distributions, especially when you have both lentic and lotic water bodies on the landscape simultaneously. I also found that lotic water bodies have a positive influence on muskrat occupancy, which is somewhat surprising at first, given that muskrats are generalists. So you may expect to see them or see a result more similar to otter and mink, in which, it's not really influential. Sure enough, there is previous research in the area that shows muskrat home ranges can be limited to these linear features associated with agriculture. And given that there's a lot of agriculture in the study area, that may be reflecting what's going on there. And then I did find that small lentic water bodies have a marginal negative influence on beaver occupancy. And that's fairly in line with what you would find in the literature, given that beavers tend to take small water bodies and make them large. So just some takeaways from this research objective here. Both lentic and lotic water bodies really should be accounted for when researching these species, especially when they're both on the landscape. And then there should be that management emphasis on aquatic factors for beaver and otter here in Southern Illinois, whereas both are going to be likely important for muskrat and mink. All right, so moving on to the second research objective here of looking at possible impacts of beaver and muskrat presence on the terrestrial mammal community. So to address this question, using the same data set collected as before, so 67 sites across a gradient of water body type and size. But here I'm actually using the occupancy probability that I calculated before for beaver and muskrat as an indicator of their presence. And then while camera traps weren't really great at detecting semi-aquatic mammals, they're great at detecting other terrestrial mammals. So here I'm actually using what is essentially the bycatch from those camera traps to quantify different measures of species diversity within the terrestrial mammal community. There are quite a few different measures of species diversity. So first you have alpha diversity, which is simply the diversity of a community at a certain site or an area. And for that, I'm using species richness, which is again simply the number of species at a site. You also have beta diversity, which is the change in diversity between sites, or the change in alpha diversity. And for that, I'm using Sorensen's dissimilarity matrix, specifically using this beta part R package. This package conveniently calculates Sorensen's, which represents overall beta diversity, but then it also partitions that out into 2 different measures. So you have Simpson's matrix, which represents species turnover. For example, if you had one site with species A, B, and C, and another with A, B, and F, that difference would be captured by this turnover. And then you also have nestedness, which is a measure of species loss. So again, one site with species A, B, and C, and another with just A and B, that difference would be captured within nestedness. And then finally, you do have gamma diversity, which is the diversity across the entire landscape. And I'm not looking at that because then I wouldn't be able to differentiate the influence of beaver and muskrat. So to actually tackle this question, I'm using those measures of alpha and beta diversity as a response in a couple of different models here. Specifically for alpha diversity, again, that species richness is a nice vector, so I can just use a generalized linear model to look at those influences. Beta diversity, though, is a matrix, so I can't use a GLM. So instead, I used a distance-based redundancy analysis, which is a form of constrained ordination, but Very simply put, it performs a regression that allows your response variable to be a matrix. I then did a backward selection function to determine a base habitat model using those covariates that I mentioned earlier, considering different linear and quadratic terms. And then I took that base model and then added in whatever variable of interest. So in this case, beaver occupancy, muskrat occupancy, or the presence of a beaver dam, and then performed a log likelihood ratio test to determine if that more complex model was a better fit. And that would be indicated by a significant p-value. And then kind of took a top model from each of those measures of species diversity and just kind of looked at the direction of influence that those covariates were having. So quick overview of that camera trap data for the terrestrial species. I captured 21 different terrestrial mammal species across all sites. Some of those, like the raccoon, were found at nearly every single site, while others, such as the eastern chipmunk, were found at very few sites. Moving on to the log likelihood ratio results here, I just have a table in which this model column is that model comparison between this base model and whatever variable of interest, and then the columns for each measure of species diversity. Bringing in the p-values from that test here, the significant values are highlighted and in bold. So sure enough, with species richness and that measure of loss across sites, adding in whatever variable of interest didn't provide a significantly better fit compared to that base habitat model alone. But when you look at overall beta diversity, sure enough, having a dam and a muskrat did provide a significantly better fit. And then for species turnover specifically, again, having a dam, a muskrat, or a dam and a muskrat did seem to provide a significantly better fit to that data. And then again, as I mentioned, bringing in this top model, for richness and nestedness, that would just simply be the base habitat model. And for overall beta diversity and species turnover, it was the inclusion of a muskrat and a dam. So looking at those model results here, an NA simply just indicates that that associated covariate was not included in the top model. So for richness here, it was just a base habitat model. So we have a positive influence of elevation and a negative influence of forest squared. And kind of a wonky interpretation, but a negative value of forest squared just indicates what looks like more like a bell curve in which areas of moderate forest cover have greater species richness and less on either extreme. Looking at overall beta diversity, muskrat and dam both had a negative influence, whereas size and elevation had a positive influence. Simpsons, which is again that turnover, muskrat and dam had a negative influence as well as size. And finally, nestedness, which was just that base model, had the same results as species richness, in which elevation had a positive effect and forest squared had that negative effect. So what does all that mean? Overall, I found that beaver presence alone doesn't seem to be influencing the terrestrial mammal community. It may be that those other behaviors that I talked about, such as tree felling and digging, are having either a minimal influence or maybe it's just not, that signal isn't strong enough to detect in my data. I also found that beaver dams have a negatively influenced beta diversity. And I scratched my head at that for a while, but really it's just suggesting that at sites with these beaver dams, you have less species turnover compared to sites without. So essentially, beaver dams seem to be attracting the same set of species relative to other sites. And that may be because these dams are creating this heterogeneous habitat, maybe acting to just attract those same set of species across areas where you find beaver dams. Looking at muskrat is similar results to those beaver dams in which muskrat presence didn't influence species richness or loss, but instead had a small negative influence on overall beta diversity and species turnover. Again, this could be due to the fact that muskrats are increasing this habitat heterogeneity and attracting those same set of species. It could also simply be correlation, given that muskrats are wetland obligates. Perhaps these same set of species that are being attracted to that area are also coming there for the same reasons. So future research would need to kind of look at the actual effect and measure the effect that muskrats are having to be able to kind of connect that dot in the middle there. Kind of wrap all of that up. Again, in areas where beavers rarely dam, it seems that their presence alone, without the presence of a dam, is not influencing species diversity. But instead, the presence of a beaver dam did influence beta diversity, but that's different than what's been found elsewhere in different areas. And as I mentioned, future research would really want to kind of focus in on measuring those direct effects that these species are having, specifically in those lower trophic communities, to draw some level of connection on how it may scale up. All right, moving on to the last and final research objective here, determining the status of nutria in southern Illinois. As I mentioned here, for nutria, we're bringing in all survey methods here. So sign surveys, camera traps, those detection canines, and the eDNA sampling. Looking first at sign surveys, camera traps, and detection canines in 2023, found nothing. Found no nutria across all 67 different sites from these 3 survey methods. And selfishly, I was a little bit disappointed because I couldn't do any cool analyses with that. Overall, that's a good thing. I talked a lot about the effects and harm that nutria can have on the environment. So overall, it's a good thing that nutria were not found at these sites. But just to be sure, we did bring the detection canines back in 2024, in which they surveyed an additional 59 sites that were all focused on having good nutria habitat, which specifically required having emergent vegetation. So again, these detection canines surveyed all of these sites extensively and still found no nutria. Again, that's a good thing. But around this time, we did receive the eDNA results back from that external laboratory. And sure enough, across both qPCR and metabarcoding, they identified 3 different sites that had nutria DNA. Looking at a map of the study area and where those sites were found, some of them were weird, some of them were what you expected. So they're fairly spread out across the area. This site on the furthest left was a small pond in a higher elevation area in the Shawnee National Forest. Was not very typical nutria habitat. Didn't have any emergent vegetation, was fairly woody. So that one was a bit odd. These other 2 are more of what you would expect to find nutria, in which the middle one was a small stream branched off of the Cache River. This one on the right was actually a small stream located around 3.5 kilometers away from that original location where nutria were trapped in 2022. But all that to say, given this, it's still fairly likely that nutria are not established in southern Illinois. And looking at this classic invasion curve, where time is on the x-axis and you have this cumulative impact on the y, it's likely with nutria that we're still somewhere low on the curve, where we may be getting these transient individuals, but altogether, some underlying biological mechanism may be preventing their establishment across decades. So it's likely to say that they're not established in southern Illinois. So wrapping everything up, what I've talked about so far, I did identify a relative difference between terrestrial and aquatic factors for semi-aquatic mammals, specifically some interesting difference between lithic and lotic water bodies. And I also found that camera traps were perhaps not the best for detecting semi-aquatic mammals. And then I also found in southern Illinois specifically, and perhaps where other areas where beavers are not damming, beaver presence alone doesn't seem to be influencing terrestrial mammal diversity. Instead, I found that dams and muskrat presence may seem to be attracting those same set of species. And sure enough, I found that nutria are not established in southern Illinois. But continuous monitoring will be pretty essential to help prevent the establishment of those species in this area. So with that, I would just like to thank the IDNR for funding this research. I'd like to thank my co-advisors, Dr. Guillaume Bastille-Rousseau and Dr. Charlotte Nahr, for all of their invaluable help over the years. I'd like to thank my committee members, Dr. Brent Pease and Mike Eichholz, for all of their help modeling, getting my proposal together. I'd like to thank the Spatial Wildlife Ecology Lab for all of their support, for answering many, many questions in the lab. I'd like to thank my family and friends for all of their support. Could not have done it without them. And I'd like to thank my wife for all of her support and putting up with me for many years. And with that, I will take any questions.